Good afternoon, everybody. So I'm going to present you the activity of PDC line Pharma. We are in the field of cancer vaccine, and we are developing a new class of cancer vaccine based on an off-the-shelf antigen-presenting cell line. In a nutshell, the company is a spin-off from the French Blood Bank, which is a very large organization in France that have the specificity as well to have academic research lab associated, and the scientific founder of the company was leading one of his lab, and he was the first one to identify a very rare disease affecting a subset of the dendritic cells called the plasmacytoid dendritic cells. And this leukemia is very rare because the prevalence, the quantity of the cells is a very few percent. The majority of dendritic cells are myeloid. And based on the blood of his patients, of his leukemia patients, he developed a cell line, GMP approved by the French authority for clinical use, but we are still using today as a vaccinal platform. So we are now a team of 33 people, including seasoned executives from biotech and pharma. We'll show you that later. In terms of funding, we get close to 56 million euros in equity and non-dilutive money. We are based in Belgium and France, in Grenoble, and Belgium in Liège. Our PDC line has 3 key advantages, and if I summarize, it's very easy to expand. It's a leukemia cells, so it can grow in suspensions in bioreactor. I will show you that later. The origin of the cells being plasmacytoid and leukemic as well provided very interesting property in terms of capacity to expand, to prime and expand cytotoxic CD8 T cells. And it's a versatile platform. I will show you. We just have to load with GMP peptide, representative of tumor antigens, and we can that fixed to the HLA present in membrane of the cells, and we can load with a lot of different antigens, and according to the antigens we are selecting, we can address any kind of liquid or solid tumor. Today, we are focusing in one indications. It is the non-small cell lung cancer. We have an ongoing clinical trial in phase 1/2 on 64 patients, and I will show you the first result, the primary result of this study. The pipeline of the companies presented in this slide. So a first academic study was done in melanoma in 9 patients. It was a dose range with the vaccine alone. It's completed. The result has been published. It's not active anymore. We have the PDC lung, which is ongoing in phase 1/2 with 6 shared antigens. And we have ongoing as well preclinical stage personalized neoantigens. So where here we look for in the tumor of the patient, biopsy of the tumor of the patient, which are the mutations that can express specific antigens that will be presented to the CD8 T cells. And we manufacture this peptide that we load to the cell line. So it's really personalized, and that is at preclinical stage. We signed a first licensing deal in 2019 in Asia with LG Chem. I'm sure you know LG for the flat screens and electronics, but it's as well the 10th chemical company in the world. And they have a life science divisions in Asia. So we provided the right for South Korea and option for the rest of Asia. You see the financial here. But at the top of the money, what was important was to have a partner in Asia and to have a validation through a partnership of the value of our technology, because this deal was signed at preclinical stage before we launched the clinical trial in the non-small cell lung cancer. The team, as I said, we have a team with strong expertise and experience in cell therapy. You see the CMC, Claude Dédry, is a former GSK guy and worked in several cell therapy in the past. Shanna De Bruyne, the medical director, managed the cancer vaccine of GSK vaccine. The scientific founder, Joel Full-Time, and myself have already involved in several cell therapy company and IPOs and M&A. One of them I sold to Genzyme, by the way. Okay, so let me show you how the technology works quickly. As I said earlier, we have this master cell bank where the cells are stored in liquid nitrogens, fully characterized and purified. And from the working master cell bank, we develop working cell banks to expand the supply. And when we want to launch a manufacturing campaign, we just have to seed single-use bioreactor. We have 1 liter, 10-liter bioreactor. We want to go up to the 50 liters in the next clinical trial. And we seed the cells with in suspensions with a synthetic medium. We use the Lonza system. And after 9 to 10 days of culture, we have billion of cells. And at the harvest, we split the cells in bag according to the number of peptide we want to load, because we will load peptide individually to avoid any competitions. Then quickly, we pull the cells in irradiate to stop the proliferations. It's a leukemia cells, I remind you. And then we put in a specific technology, it's a vial of aseptic technology with a cryoprotectant. And like for the COVID-19 vaccine, we just have to ship in minus 80 degrees fridge in the hospitals. And when the patient is ready, you just have to thaw the cells from the vial and with a syringe to take it and infuse IV and subcute to the patients. And the vaccine will make the job, which is to go to the lymph node to prime CD8 T cells, naive CD8 T cell that will become memory cytotoxic, migrate to the tumor microenvironment, and what we expect to squeeze the tumor. So this process has been effectively done in more than 12. We have produced more than 12 batch, and we have more than 90% of success of release. So the CMC is completely under control in our case. So let me show you now the first results of the current clinical trial on non-small cell lung cancer, which is ongoing. We have the design of the clinical trials, as I said, is on 64 patients. We have 4 cohorts of patients because we have two doses of vaccine we are testing, 2 million cell per peptide and 20 million cell per peptide, and low dose and high dose. And we test first with the vaccine alone in adjuvant setting. So here the patient can have a surgery. In general, they have a chemotherapy adjuvant to remove residual disease. And after washout period, we infuse 6 times weekly the vaccine, IV and subcut, 6 patient low dose, 10 patient high dose. Then B1 and B2 are the second populations. It's here metastatic patients, naive to the checkpoint inhibitors, so they are diagnosed with metastatic non-small cell lung cancer, so they cannot have surgery. Right away, the standard of care is pembrolizumab, and we are in associations with pembrolizumab with the vaccine. Okay, and we infuse 6 times, 6 patients low dose, then 42 patients high dose. Why 42 is because it's in this last cohort we would like to observe statistically a difference versus pembrolizumab alone. We have already completed the cohort A1, A2, and B1, and we are completing enrollment of the B2 that we expect to finalize completely before the summer. So let me show you the first result of the 3 first cohorts. Let's start by the immuno results. So we take blood sample of the patients before infusion of the vaccine, then post-infusions, and we have 3 time points, 1 week, 4 weeks, and 10 weeks post the last infusions. And you see here the result by fax dextramer directly ex vivo. So we don't amplify the CD8 T cell present in the blood. It's really what you see here, what is in the blood. And pre and post infusion of the vaccine. I don't know if we can, no, sorry, I don't have a pointer, but anyway, in the pre, what you have cohort A1 is the low dose vaccine alone, A2 is the high dose vaccine alone, and B1, it is the low dose with checkpoint inhibitor. And as you can see, we have observed expansions of CD8 T cells already on the A1, which is higher and with a higher number of responders in the A2. And we see as well a good expansions of the CD8 T cells in associations with checkpoint inhibitor. By fax, we can follow what are the memory T cell, the phenotype memory or naive T cells. And we have seen that post-infusion of the vaccine, all the responders have memory CD8 T cells specific to the antigens of the vaccine. We see better here the presentation of the data. You see the fold increase from baseline. Red is the A1, once again, vaccine alone. The green, it's the high dose, and you see well here the dose range effect, so that was expected. In purple, it's the low dose with the checkpoint inhibitor, and here again, you see the synergetic effect in immuno with the checkpoint inhibitor. So that's interesting as a first result of our current clinical trial. Interestingly, we had one of the patients that has a flu, infections, regular flu, and we have a positive control in the immuno. We follow some antigens basal antigen that are present in any patients, like the flu. And as you can see, the expansion after the flu infections in this patient is around 50, and for some of the patients, we reach this level or even above the level, which is once again quite interesting and promising for the vaccine. Okay, let's pass now to the first clinical result. So here are the targeted benefit we have for the clinical endpoints. We are following the overall response rates, the tumor shrinkage, and the progression-free survival at 9 months. You see here the results, the keynote 042, it's for the benchmark, which is the pembrolizumab alone, 39% and 43%, and we want to increase by 15% this level of 2 endpoints, so to reach 54 for the ORR and 58% for the PFS 9 months. So what we can see for the B1, which is the 6 patients of the low dose, is what I'm showing here. So we have 6 partial response. So we call partial response in green when you reach more than 30% of tumor shrinkage. And as you can see, for the partial response we have, it's not borderline, it's really deep partial response. We have 1 stable disease and 1 progressive disease. But for all of the patients, we had in the best overall response, a squeezing of the target tumor. When you see the evolutions of the tumor over the time, here you see the months versus the tumor shrinkage. You see that the duration of the effect is quite good on this first population of low dose with the 6 patients. We are up to more than 15 months for one of the patients, and with good result and good trend that are still ongoing. We did as well on the first 6 patients a kind of correlations between the immuno and the clinical response. We call responder when you have partial response, complete response that we don't have yet, and stable disease. So when the patient stays stable for a long time, and we correlated with the immuno response, and we have observed that the big majority of the responder have immunological response, and the one, for the moment only one, did not have any immunological response. So, which is an interesting first sign of correlations that we hope to validate, of course, with the 42 patients of the B2 cohort. That's my last slide. So just in terms of timeline, we should get the interim clinical study report with the 19 out of the 42 patients of the B2 available with the PFS 9 months and the ORR in July, August. So we expect to publish this data or to present in conference, and to have the final clinical study report end of Q1 2024. We are financed to finance the current clinical trial, but I'm already preparing the next one. We hope to do a phase 2 or phase 2/3 randomized study after this clinical trial, so it's starting in 2025, and at this time we'll need, of course, significant amount of money, and in parallel to address new indications outside the lung, and to set up in US and Asia. And I believe it's my last slide, and I would like to thank you for your attention, and I don't know if we have time for questions. If not, just afterward. Thank you.